I feel like saying, and now for something completely different, since we're going to be talking about the microbiome for the next 10 minutes. So let's talk about the 90% of you that is microbial. The average adult human, 90% of your cells are microbes. They're much smaller than your cells. Most of them live within the intestinal tract, but they're also colonizing your skin, upper respiratory tract, and basically cover many diverse ecosystems within the body. These organisms provide a number of critical functions for you day in, day out. They're essential for your early maturation and development, particularly the immune system, but also with ongoing digestive functioning. They actually produce hormones and neurotransmitters. They've been doing this for billions of years. So it's not surprising to me that in our evolution, we have come to use molecules that they have long been producing as part of their normal metabolism and physiology. Whenever you sit down to take a bite to eat, not only do you digest, they digest as well. So they're a critical part of our metabolism and certainly influence our own physiology and metabolic functioning. We thus consider them sort of the great unknown frontier that is a druggable target and a way to assess disease risks and new therapeutic interventions. But to get there, it requires that we find causal effects of the microbiota and clear molecular mechanisms of action. Within the center, this is our goal. We have a focus on function, myself and my other fellow faculty members. We frequently start with a patient cohort to understand, is the microbiome playing a role? If so, is it for everyone? Is it for particular patient populations? We use sophisticated computational methods to look at the longitudinal dynamics of the microbiota, how it responds to perturbations, how it is changing before a patient necessarily has a particular disease event. With the signals we extract from these initial analyses, we move into experimental models. They could be in a test tube. We use germ-free mice with defined colonization, small molecules, changes in diet, et cetera. We also use large animals in some case to model an omnivore digestive tract. With what we learn from these in vitro and in vivo studies, we're ready to return to the bedside and ask what can we do as a potential therapeutic intervention, particularly if we can show what we may have learned in an animal model definitely translates to human populations. So I'll give one example in food allergies, how we've been able to use the microbiota to provide a cure for this disease that affects many Americans and people worldwide. If we look at the atopic march in the development of food allergy, it starts with food sensitization, frequently very early in life. This leads to the development of an allergic or Th2 cellular response within the mucosal T cells, which tells the B cells to make allergen or food component specific IgE. This results in the recruitment of mast cells into the bowel wall, and with re-exposure to the foodstuff can produce a life-threatening and anaphylactic reaction. So if we look within the gut mucosa, tolerance to these food allergens versus the development of allergy, we actually have a very specific corridor from the microbiota. Certain microbes are stimulating dendritic cells, talking to the T cells, talking to the B cells to enforce tolerance to food antigens. In contrast with allergy, the microbes are sending signals that cause the dendritic cells to present this as something foreign and lead to the production of IgA and ultimately food allergies. So we've been able to show with certain microbes we can convert these phenotypes from allergic to tolerant. This is an area of unmet need. These patients do not have an option for a cure. It is avoidance, it is painful desensitization regimens. So being able to give them a therapeutic option really can change the field and basically life for these patients and their families. We've also found by understanding the molecular mechanisms by which we can convert an allergic reaction to one that is tolerant, this is opening opportunities for us to approach other atopic diseases, such as asthma, such as atopic dermatitis, or even autoimmune diseases where instead of a Th2 response, maybe it's a Th1 or a Th17 response that we wish to convert. We have patented multiple therapeutic consortia for food allergy. These have been licensed to Pareto Bio. Nuri Hong, the CEO of Pareto Bio, is in the audience, and he and I are happy to talk to folks afterwards. As I said, we've taken this down to molecular mechanisms of action. We know within the T cells we are targeting two pathways, MyD88 through toll-like receptor 2 and ROR gamma T. We have further small molecules on the microbial side that we know are modulating epithelial responses and are modulating intermicrobe responses within the gut lumen that can also promote a tolerant or regulatory phenotype. Just to show some of the data, this is our animal model using a well-published mouse model with a defect in the interleukin-4 receptor that makes it trigger happy. Mice are sensitized for a period of 8 weeks, after which they will anaphylax if they're exposed to ova or peanut butter or particular milk allergens. And as you can see in the data on the left-hand side, this is showing temperature drop along the y-axis. During anaphylaxis, your body temperature drops, and this is showing over the first 60 minutes. You can see along the top, if mice either weren't sensitized or received our therapeutic consortia, they converted from clinical anaphylaxis to not having any clinical symptoms. So this was the first indication that we were having therapeutic efficacy. As you can see on the next two plots, the first or the middle one shows total IgE, the one on the right shows allergen-specific IgE. We've converted the mice from having very high levels of allergen-specific IgE to near undetectable levels. So we've actually gotten rid of the pathogenic molecule. So this is giving us a very high resolution molecular understanding for how these microbes are working within the body. Within the center, we have additional IP that we've developed for other disease states, including how the microbiota modulates the gut nutrient environment. We use this to develop a monobacteria therapeutic for C. difficile. We chose C. difficile because it is the one disease where we are absolutely certain that the microbiota is important. For patients who can't clear this infection from antibiotics, they're frequently given fecal microbiota transplant, a complex microbiota which has high efficacy but has significant risks in immunocompromised and other very ill patients. There have been causes of death. We were able to get it down to a single microbe that we found could outcompete C. diff. 6 complex mechanisms of action, but all defined to a molecular level. So now we are able to take FMT from many hundreds of organisms to this organism, which is genomically characterized. We know how it operates. We know how we want to deliver it. Having defined the platforms to be able to undertake these types of questions, we're now applying them to other areas. We're looking at the role of the microbiota in metabolic syndromes. As I said, as you are digesting, your microbiota is digesting day and night. So by understanding what these molecules are, we can understand how they influence a healthy person. How do they influence somebody that's at risk for diabetes? We found small molecules in the microbiota that raise blood pressure. So for hypertension, there are potential therapeutic opportunities. And as I said at the beginning, microbes produce hormones, neurotransmitters, and other molecules that interact and interface with our tissues. So this gives us opportunities to think about whether it's the gut-brain axis, how the microbiota is modulating these effects. So just to give an example, this is some data showing how we can use different microbes to alter how you are metabolizing glucose or lipids, whether you are awake and eating or while you are asleep. So this gives an example that the microbiota is a credible entity to approach. We have to understand the mechanisms of action to take it to the next step. Is there an opportunity for how we're going to use this clinically? As I mentioned, the food allergy therapeutics have been licensed to Pareto Bio. The company has since developed a proprietary lead therapeutic that it is taking forward to clinical trials. It is in the midst of the Series A opportunity, and as I said, myself and Nuri Hong, the CEO, are happy to talk to people about this opportunity and what our plans are. I am the scientific founder of the company, and I also chair its SAB. And through the Host-Microbiome Center, we have additional IP filings related to atopic diseases and related to other areas. And I'm happy to talk about what our faculty in the center is doing to further promote use of the microbiota in clinical medicine. Thank you.